In this video, we will discuss about the effect of CAR design in NK-92 functionality. Here in the panel A, we have seen the three different kind of CAR design, or basically there are two different generations. First one is the first generation that has only one signaling domain, that is CD3 zeta, and which is also found in the endogenous NK-92 cells transmembrane region, and it is connected with this single chain fragment variable using hinge. Hinge is basically made up of CD8 alpha region. And the second one is the second generation CAR, where extracellular region is similar. The transmembrane region is CD28, which is not endogenously found in the NK-92 cells, and there is a signaling domain, that is CD3 zeta. Similarly, the another second generation CAR is this, which has this single chain fragment variable, and the hinge, which is made up of CD8 alpha, and the transmembrane region, which is made up of CD137, and it is also a intercellular co-stimulatory signaling domain. Along with that, the signaling domain, that is CD3 zeta. So these are three different kind of CAR construct that are being evaluated. So first they checked the endogenous expression level of the CD28 and CD137 in the NK-92 cells, and then compare them with PBMCs. PBMCs is a control where these are found to be expressed in the high amount, and we can see that in the NK-92 cells the expression levels of CD28 and CD137 is very low compared to the PBMCs. This confirmed that the CD28 and CD137 are endogenously expressed low compared to the PBMCs. Thereafter, they checked the expression levels of the CD28 and CD137 in the lentivirus transduced NK-92 cells of three different CAR construct. So since CD3 zeta is already found endogenously in the NK-92 cells, if their expression levels is high because of the combined effect of endogenously expressed and expressed because of the transduction. And this we can see in the reducing form and also in the non-reducing form, compared to the expression level of CD28 zeta and in the reducing and non-reducing form. And since CD28 zeta size is higher because it combines both CD28 zeta and the CD3 zeta, we can see its size is higher compared to the CD3 zeta alone. And CD137 also expression levels are also shown here, but in the non-reducing conditions their expression levels are significantly higher, or seems to be higher compared to the reducing level. Another interesting things that they have seen is that this CAR construct, this first generation CAR, the first generation CD3 zeta CAR construct is expressed in dimer and tetramer positions also. Here it is shown as the dimer positions. So, and this results for the panel C is from the anti-CD8 alpha immunoblotting, indicating that antibodies that are used to detect this are anti-CD8 alpha because it is attached to the intercellular domains, and then based on the size they predict that this could be anti-CD28 signals and anti-CD137 signals, and not just based on the size but based on the informations that these cells are transduced with CD28 CD3 zeta CAR construct and CD137 CD3 zeta CAR construct. So from this results, and also mentioned in their text that since this CD3 zeta is endogenously expressed, some of the cells they are expecting to see more incremental functionality in this first generation. However, because of the absence of the co-stimulatory domain, it does not show much high functionality in some kind of cells. And the researchers finally also made a point, or mentioned a point, that NK-92 functionality against the target cells depends on what kind of target cells are used, and this needs to be optimized for different target cells separately, because of the inherent toxicities, ability of the NK-92 cells to detect target cells. So this is what they finally conclude in this figure. If you need to understand more about this effect of the CAR design in NK-92 functionalities, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.